Welcome everybody, my name's Rachel from Bats Without Borders, and I just wanted to give you all a warm welcome to our second Women in Bat Science Indaba. For those of you who are tweeting or on X, our hashtag for today is WiBS2024, so please make sure you do that. And today, this event is in celebration of International Day for Women and Girls in Science. And this quote taken from the UNDP website is obviously, we understand that gender equality is a global issue, but women and girls represent, of course, half of the world's population and therefore also half of its potential. So gender equality, besides it being actually a fundamental human right, is essential to achieving peaceful societies with full human potential and sustainable development. So I wanted to have a really big thank you to our sponsor for today, so Wildlife Acoustics, who also supported our event last year, and also a big shout out to the Rufford Foundation, who have funded many conservation projects across developing countries and support a lot of women working on conservation projects. And myself and Helen Taylor-Boyd from Bats Without Borders have both received funding for our PhDs and are incredibly grateful. So if you haven't come across them before, I'd highly suggest you check them out. So today we have a fantastic lineup. So I am delighted. I'll be introducing shortly our keynote speaker, Dr. Katendi Changula from the University of Zambia. And she is from the School of Veterinary Medicine. I'll then be handing over to my co-host today, Dr. Elisa Fuentes Montemeo. So Elisa is one of our Bats Without Borders board members, and she is also a senior lecturer at the University of Stirling. And for those of you who are interested in finding out more or getting involved in Women in Science, so we have a sign-up sheet. So Ruth will kindly pop in the chat this link, and if you can sign up to that, any of other events that are happening around Women in Science, we will be able to share that with you if you sign up to that mailing list. And a big thank you to those who donated for today, and thank you very much. These donations help us to keep these events running and to ensure we can also always have some free places. But if you're in an opportunity and would like to contribute to our work in the future, we'd also recommend you look up becoming a friend of Bats Without Borders. This is a great way to support our ongoing work. So I'm just going to share a video from Wildlife Acoustics, our sponsor today.
Wildlife Acoustics is always working, step by step, to improve acoustic research techniques and song meters to help enhance wildlife research. There are a multitude of ways to use song meters to understand the world around us more intimately. However, there is one general reason listening to the world around us is important, because listening begins with concern. The passion and concern for the environment and wildlife is at the heart of all the work at Wildlife Acoustics. And as a wildlife biologist, it becomes clear as song meters are used for ecotoxicology research in the depths of the Belizean jungle and bat species identification. We care, we listen, and we share a mutual interest with our wildlife and environment.
Welcome Dr. Katendi Changula, who is our keynote speaker. So today's talk is going to be about Katendi's journey in bat research in Zambia. So navigating pathogens and ecology. And as I've mentioned, there's a lot of information about Katendi's work and our other speakers today in the program. So Katendi is based at the University of Zambia. She's in the School of Veterinary Medicine, and I'm delighted to have her as our keynote today. Thank you, Katendi.
All right, thank you very much. And thank you. Such an honor to be invited to be the keynote speaker at such an event. So I'm really going to talk about some of the scientific research we've been doing with bats and somehow also weave in some of my journey into actually working with bats. And even if there is just one name here on the title page, there are a lot of people who I've worked with, so many that I could not put them here, but I do have a couple of slides at the end that highlights everybody who's been part of this work. So I'm going to start with a background of pathogens, because I'm coming from the pathogen aspect of bat research. So I work looking at emerging and re-emerging zoonotic diseases. And one of the diseases which is my main focus, or one of the virus families that is my main focus, is the filovirus family. And in this family, actually, we have several genera, but there are two that are of main interest because these actually cause disease in humans and non-human primates. So we have two genera which are important. So this is the orthomabic virus and orthoebola virus. You will forgive me if I go back to their old names, because these are just new nomenclature that just came up towards the end of last year. So I'm still trying to transition. So we have these orthomabic viruses and orthoebola viruses, and they have been found in bats, and they cause disease in non-human primates and man, and most of them are endemic to Africa. So as I've mentioned, they cause disease. So the diseases are known as Marburg virus disease or Ebola virus disease, and I'm sure we are all familiar with Ebola virus disease, and some of us may be familiar with Marburg virus disease as well. So these are basically among the most lethal human pathogens. They cause what is known as a viral hemorrhagic fever. And the reservoir host for orthomabic viruses is known, and is known to be the Egyptian fruit bats. But the reservoir host for orthoebola viruses is still not known. So geographical distribution, when you talk about Africa, with the Ebola viruses, they are approximately within 10 degrees north and south of the equator, but the Marburg viruses are more widespread and come down to Eastern and South Central Africa. And then we also have the Asian and the European filoviruses, which are not pathogenic to man. So I just want to show a map of all the showing all the outbreaks that have happened in Africa. So highlighted, the shaded in gray are the countries that have had outbreaks, and these other ones in this peachy color are countries where you've had people actually taking the infection after being exposed in an outbreak country. I come from Zambia. So this is Zambia here. And if you look at Zambia, half of Zambia's neighbors have actually had filoviruses. But why is Zambia seemingly clean? We have never had a recorded or reported outbreak of any filovirus disease. And this brings the question, so why would I look for filoviruses? So this map is one of the answers, because if there are filoviruses here, there probably are filoviruses here. So I was introduced to filovirus research, to bat research, really in a roundabout way. As I said, I'm coming from the pathogen background. So when I was doing my PhD was in filoviruses, but in diagnostic tests. So nothing to do with bats. But I needed funding for my work, and my then supervisor introduced me to his collaborators who were actually looking for filoviruses in bats in Zambia, and were willing to fund my PhD program. So basically, I had to join them in the bat part of the research because I was now part of the team. And so that's how I got involved with bats. And as I said, strictly, I was looking at bats from a pathogen perspective. So when we look at bats and filoviruses, of course, I've already said that the reservoir host for the orthomabic viruses is known to be the Egyptian fruit bats. But for the Ebola viruses, it's still out there. We do not know, but they still do suspect that it's bats. But even if bats do play an important role in the transmission of filoviruses, there really may be other animal species that are involved. For example, duikers and non-human primates, although non-human primates are susceptible hosts like humans. But then there are other animals. For example, when you look at the Reston virus, which is from Asia, pigs were involved somehow in the chain. Dogs have been known to seroconvert in Ebola virus outbreaks. Rodents live, some of these dwell in caves where these bats dwell, and there's a whole ecosystem within that cave. How are the rodents also involved within the ecosystem of these filoviruses where they are found? So it's not cut and dried. And then when you look at the distribution of filoviruses, it really is wider than we thought. Not maybe, it really is wider than we thought, because we are continuously having either new species or new strains, discovery of new strains, or new locations. So for example, in 2023, we had the first outbreak of Marburg virus in Tanzania. Tanzania had never had an outbreak before. So continuously we're having this happening. And also you find that probably there may be some misdiagnosis or underdiagnosed cases in countries that have had no history of an outbreak of this virus disease. So for example, some studies have shown that in countries that have had no recorded outbreak of Ebola virus disease, both humans and non-human primates have high antibody or seroprevalence to Ebola virus. And how is that possible? Is it that there is Ebola viruses that are non-pathogenic? Or is it that there are other filoviruses that are antigenically similar to Ebola virus, but with lower pathogenicity? So there's so many things that we do not know about filoviruses and the dynamics around their transmission. And basically these populations that have this high seroprevalence, it means that they are highly exposed to this virus in one way or the other. So when we divide the filoviruses into the Ebola and the Marburg, if you look at this predicted ecological niche of Ebola viruses, of course, Zambia is here. As I mentioned, 10 degrees north and south of the equator, so it's more around Central Africa. So with the Ebola viruses, really the risk to Zambia or having an outbreak originating from Zambia is relatively low. But when you look at Marburg viruses, as you can see, the risk is higher because if you look at the niche of ortho Marburg viruses, it is quite a wider distribution in Africa, coming down into Southern Africa as well. So now with that background of why would we in Zambia be looking for filoviruses, I'm going to talk about some of the research that we've been doing, but I'm going to concentrate on two bat species, and that's Eidolon helvam and Rosetus aegypticus. So these are the sites in Zambia, these highlighted in red, and plus the Kasaka, the famous Kasaka National Park. So these are the areas that we have actually conducted bat research in. But of course, I said I was going to focus on two bat species. So first of all, we'll talk about Eidolon helvam, or the straw-colored fruit bat. So this is famous because of the annual migration to Zambia, which is known as the biggest mammal migration in the world. So these annually migrate to Zambia around October to January each year, and mainly found in Kasaka National Park, but they are also found in other areas like in the suburbs, in Ndola, Lusaka, Kabwe, Kasama. And even if the period of migration is in October, out by the first week of January, surprisingly last year, I was able to see a colony in April in Lusaka. So that's a bit strange and a bit worrying. That's why that is happening. So where we actually collected bat samples from was from Kasaka National Park as well as from Ndola. So this is just a picture of the bats as they go out to forage in the evening. The interesting thing about the bats, especially Fondola, is originally they just used to come to one street in a specific suburb. And over the years, the colony has grown and they're basically come through to the entire suburb in that area. So we conducted some tests on these bats over a period of years, and we never found any viral material or any viral genome. So basically the bats, when they came to Zambia, did not have any virus with them, but any filovirus. But what we found was exposure, evidence of exposure in terms of antibody or seroprevalence. And the interesting thing is that the seroprevalence or the variety of antibodies that were there would change from year to year. So you'd have different proportions of the different viruses represented over the years, and it was never uniform. Some antibodies to some viruses were never found in some particular years. And also, for some viruses, it would be interesting to note that where you find high seroprevalence in the bats, there was also an outbreak around the same time. So for example, the Sudan Ebola virus, you can see 2013 was the most prominent. And if you see 2012, there was an outbreak. So some of the viruses actually had that kind of pattern or picture. So about 9% of these bats actually had filovirus antibodies. So there are several things that we might, several ways we can interpret these results. We could say, okay, maybe these bats are not reservoirs, but they are frequently exposed to the spillover of the viruses from other reservoir hosts within Central Africa, from where they come from before they come to Zambia. So they are exposed there and then lose the infection and are just seropositive when they come to Zambia. Or these bats could be a reservoir, and the seroprevalence is just the way the seroprevalence of the different filovirus species changes over the years is just a reflection of the shift of the proportion of the different filoviruses that are being maintained in this bat population. Or these antibodies that we are detecting are basically from unknown filoviruses, but with similar antigenicity to the species that we know. And we can also hypothesize that we may not be able to detect all antibodies because there may be some difference in antigenicity of some of the viruses that are more distinct from the known species that cannot be detected with the test that we are using now. So basically, these bats, at some point in their migration, they encounter the virus. But the question is, where do they encounter the virus? So this is just a map, IUCN map, to show the distribution of these eidolon. So it doesn't mean that the eidolon migrate from West Africa all the way to Zambia. Of course, they've got different migration patterns, I'm sure, depending on the colonies. But there is a point where they encounter the virus. But when we look at other studies that have looked for the virus in these eidolon in different countries, they also don't find the virus. They only find the antibodies. So that's why it is not enough just to look for the virus. We need to find what is happening within the bat population, where the bat populations are found, and the animals are surrounding the same bat populations. What is happening in that ecological system that is causing exposure to these bats? So it's very important, really, as we are coming from the pathogen side, to not only look for the pathogens, but also to actually partner it with ecological studies to understand what's happening and how the bats are getting exposed to filoviruses. So that is it for eidolon. So we haven't done any eidolon health sampling since just before COVID. And this is basically because we were getting the same results over and over again. So looking for anything within the Zambian context, I don't think is useful, and we need to explore further from where in different different countries that these bats may be coming from. So I'll look now to the Rosetas aegypticus, which are known reservoir hosts of the orthomabic viruses. And just briefly, the aim of our study is to understand the ecology of this orthomabic virus outside endemic countries. So back to this map again. Again, Zambia has never had an outbreak, but of course, looking at what's happening around Zambia and all the other things that I've mentioned before, we know that we should be able to find orthomabic viruses in Zambia. And this is just a picture of an Egyptian fruit bat from the IUCN website. So the study is Leopard's Hill Cave. This is in Lusaka province in Zambia. This is just an infrared video of bats as they're emerging. So this bat cave really, of course, apart from us scientists who enter the cave, we also have the locals who enter the cave for guano. As you know, guano is very rich in nitrogen, so a very good fertilizer. And we also have tourists who enter the cave. So a search on YouTube, if you just put Leopard's Hill Cave on YouTube, you'll come up with several videos of people who have taken videos as they have gone into the cave. So this cave is used by quite a lot of people from different walks of life. So we use a hub trap to catch the bats from the mouth of the cave. And so with what we do with our research, we capture the bats. So we have sacrifice and we have capture and release. So capture and release, we will take swabs. Sometimes we'll take blood. But as you know, it's so hard to collect blood from a bat, from a live bat. And then with a percentage of the bats, a small percentage we sacrifice and collect blood and organs. So basically what have been our results with the serology? So looking for antibodies to filoviruses. So over 50% of these of these Egyptian fruit bats had antibodies to different filoviruses. But the interesting thing was over 40% had antibodies to Marburg viruses. And one thing that we noticed over the years was that there was always a peak of seropositivity around November to December each year. And then it went down in the other months of the year. So a peak of antibodies around that time of the year. So because we were not finding the virus, but we were finding the antibodies and in such high levels, we looked at the peak and we know that we have, when a bat is infected with Marburg virus, there's a peak seroprevalence 28 days post-infection. And then these antibodies rapidly decline over the next 2 to 3 months. So what we basically did is we counted about 3 months back from the peak of seroprevalence, and that was around August. So our hypothesis was that we would have Marburg virus between August and October. And we then targeted our screening to that time of the year. And truly we found September 2018, we were able to get 2 positive bats. And then October 2022, another 1 positive bat within this period that we had targeted. And what is known really about Marburg virus is that the juvenile bats are the ones that normally shed the virus. And this was basically true to what we found as well. So one very interesting thing about these viruses that we detected in 2018 and 2022 was that the 2018 was very highly similar to a DRC strain, while the 2022 had high similarity to Zimbabwe strain. But these are coming from the same bats, from the same cave, from the same system. How do they have two different strains of Marburg viruses circulating and from so widely different geographical areas? And then also the other thing is that within this cave, we not only did we detect Marburg virus, but quite a large number of viruses have been detected, some of them novel viruses. For example, this one, which was named leopard seal virus, as well as some spirochetes, the Borrelia. So many different pathogens have been found in this cave in these bats. So basically, this cave is a very suitable environment for pathogen spillover within the bats and whatever is within the ecosystem of that cave. So now, because of the results that we got with the two different strains from wildly different geographical locations, we really wanted to understand how is it that this can be. So we had to then look at the host, which is the bat. And we did a biologging study and collaborated with some researchers from Doshisha University in Japan. So basically, what did we want to answer? What is the foraging area? So this is the location of the cave. This is the location of the capital city, Lusaka. This is the border between Zambia and Zimbabwe. This is Mozambique. So the question is, the transmission risk area around the cave, how widespread is the area under the cave at risk for Marburg virus? Because we did find the Marburg virus. And knowing that within the radius of 50, within the radius of 50 kilometers, we have a very highly densely populated capital city. And is there transboundary movement, considering that we found virus that was similar to a virus that came from Chinoy caves in Zimbabwe? And then looking at the other viruses that we found, the other Marburg virus was from an outbreak in the northeastern part of DRC. And you can see that the distance as the crow flies between there and the cave is approximately 2,000 kilometers. That is very far. We also found another virus, Kasekoro virus, which was found in Uganda, but found also very highly similar strain found in the caves in Leopard Seal Cave. So how is the long distance migration? And is this a seasonal migration? Is it that the bats migrate seasonally? What is it that is happening within the bat population? So we had both short-term and long-term tracking. We put data loggers on the bats, GPS data loggers. So short-term tracking to understand foraging territory, and the long-term one to observe transboundary migration pattern. So basically, these are the data loggers, which we put on the bats. So just want to show you a short video of one of our results of the short-term tracking, and shows some interesting behavior from the bats over a period of 7 days. So each color is a bat. So the cave is here, and they go out and they come in. But some bats, actually, you realize, do not come back immediately the same day. So some bats will stick around in some other areas and then only show up. For example, I think it's, so here there's this bat which just showed up here. Probably had spent several nights in this area and then came back to the cave on day 3. And you can see that even the dispersion of the bats is quite uniform. They go in all directions outside the cave. And you see that some bats actually have a kind of character. So if you look for the orange bat, this orange bat is kind of a loner, hangs around in different places, does not go back to the cave the same night, goes back to the cave after he misses home for some time. And then, so it was really interesting to see that the bats really have different behavioral characteristics, really, as they go to forage. And the other thing that we noticed is that the bats, a lot of the bats were coming to the Zambezi escarpment, which is along here, Zambezi escarpment. And so we actually thought there were 3 main points where the bats came in the escarpment. So we thought there could be caves. And because some of the bats would spend a night or 2 before coming back to the Leopard's Hill Cave, so we thought there could be caves where these bats are also interacting with other bats. And one thing we also were able to determine was the total flight distance was about just under 100 kilometers a night. So the radius was about just under 50 kilometers. So a total of about 100 kilometers per night. And then, of course, with the capital city being here, we could see some of the bats coming towards the capital city. So we then, with GPS location, we went to the escarpment and were able to locate one of the points. 2 of the other points were in areas that we could not access, they were inaccessible, but we were able to locate one of the points. And what we found actually was not a cave. What we found was a crevice. So we're like, okay, so these bats may come down all this way to forage and find that it's too late to fly back and hang around here for a day or 2 and forage here until they decide to go back to the main cave, because it's really not a place where we didn't find any bats in there or any really smell of guano that could tell you that bats are resident there for a long period of time. And another interesting thing that we found was that there was a difference between the seasons. So April is the end of the rain season. The fruits are all gone. And what we found is that the bats forage over a wide territory and they go into the remote areas to find their fruits. But in October is towards the end of the dry season, but fruits are already emerging. And what we found is that they foraged over a smaller territory. The radius was quite small, and they foraged in homesteads. So this is a homestead, and we have the bats concentrating. This is a tree, this is another tree, could be another tree here. And what we could see is that these are probably mango trees, because most people will plant mango trees around their homesteads. And remember that we found virus transmission, virus excretion in October. So we were able to detect the virus in October, meaning that even as these bats foraging movements change, or foraging habits change, even the risk of disease transmission differs between seasons. In October, the risk of transmission to the human population is higher because these bats are actually foraging around residential areas. So there is a higher risk of transmission of the virus at that point. And with the long-term tracking, we're able to see transboundary activities. So all these yellow lines are bats flying, and this is Zambia, this is Zimbabwe, this is Manaples National Park. Of course, bats don't know borders. And so they don't know that they are crossing into, this is basically their territory. So we actually were able to see that these bats were flying over into Zimbabwe. So that is also interesting, especially that we had the virus that was very similar to Zimbabwe strain. So what can we conclude with this ongoing Marburg virus study? So the Egyptian fruit bats in this cave host multiple strains of Marburg viruses. We've seen that the foraging habits vary with the season because of the availability of fruit, and this may also cause an increased risk of disease transmission. Our results also show that there may be communication between different bat populations, and that when they communicate, they are able to transmit viruses between each other. The study is still ongoing. So in July, we put some more data loggers, and also last month, we put some more data loggers. So we just want to see the picture over different seasons. Someone is helping us to have to do home range analysis. We have a PhD student who is analyzing guano from the cave. Because remember, I said it's not enough to just find the pathogen. You find the pathogen and then what? We need to understand everything that is happening around that pathogen. So there's a student who's collecting guano. We're going to test the guano for filoviruses. We're going to test the rodents from within the cave as well from outside the cave and see whether they have been exposed. Is there any seroconversion? This will help to clarify some type how the transmission of the virus can happen between bats and humans, and also bats to bats, because we really don't know how the bats transmit to each other. And then also trying to do some population genomics of the Rosetta's bats to see how related different populations are with each other. So we are actually looking for people to collaborate with this population genomics, because we need to get bat DNA from various parts of Africa. That will help to give us a bigger picture on how these bats may be communicating, even if they are not known to be migratory bats. But are they actually one big population, or are they several different populations? So this is very important. And finally, as we celebrate women in bat science, I'm going to show you a few pictures. So this is in the field with our team, different parts. This is in DRC. This is at Leopards Hill Cave. This is at a place in central Zambia. Another picture, this is at Leopards Hill Cave, DRC. But one notable thing that you can see is where are the women? I'm the only woman there. And this is quite disappointing. Bat research in Zambia is what I can say is a new field. We have had a lot of international researchers coming in to do the research, get the results, and they go out. But when we talk about Zambian researchers, local researchers, the only other female or woman researcher that I would know is Helen Taylor Boyd, and I'm sure she's there in the, I'm sure she didn't expect this, but I've learned so much about bats and bat research from her. She's an ecologist, so she looks at bats from the other perspective, not from the pathogen perspective. And she's done so much work with the bats. But us as women in Zambia, we are just me and her. And I hope that a lot of, we tried to advertise this as much as possible. So I hope a lot of female Zambians have also joined in to listen to the talk and listen to the other talks that are going to come from the other different researchers, and will also be motivated to think about a career in bat research. Finally, as I said, I could not put all the names on the first page. So we have had so many people working with us, so many collaborators. So these from Zambia, DRC, Congo DR, and also Japan. So all these people, one way or the other, I've worked with, and still working with, and involved in bat research. So thank you very much. I hope I have held your attention.
Thank you very, very much, Katendi. That was fantastic to see an overview of the work that you are doing. So just to let you know, if you have any questions, please pop them in the chat. But I'm just going to kick off with a question. I was just wondering, I think you've sort of captured it there at the end, the really importance of interdisciplinary work. Like, I'm sure there's a lot of psychologists and conservationists who are actually quite jealous of your bat tracking data, and I think that there's a real opportunity for us to kind of work collaboratively more. So I suppose it's just kind of your advice on how do you think we could be better, working in interdisciplinary to support each other's research?
Yeah, that's a very important question, because as I said, when I first of bats and pathogens, and that was it. But then I got to learn about bats and how they're important to the ecosystem. And then also, if you find a pathogen in bats, you cannot eradicate all the bats. You still need the bats. So you need to understand the ecology around that, so that you are able to then advise the individuals living around that area and say, okay, this is what you need to do to protect yourself. So this interdisciplinary collaboration is very, very important, and it's very key. And I think it just comes out of out of interest as well, and also exposure. A lot of people actually do not think about, they only think about bats as a nuisance. And with the work, especially that Bats Without Borders is doing, and the work that also Helen does, going around and giving talks at schools and stuff like that, talking about bats, it actually shows the importance of bats. But also, when we talk about, I'm going to talk about the African setup as well, because we have a lot of people who come in. I'm not saying that it's wrong. We have a lot of people coming from outside, from the global north, and do their fancy research and go out and take it back and publish. And there is really no benefit to the local researchers. So I think us as local researchers also need to be proactive in linking to each other within the countries. So for example, I work with some researchers in DRC. I've been helping them to set up something similar to what we're doing here in Zambia. And that is a very good collaboration. So I think it's also about reaching out and having a landing page or something where we know who is doing what. So if you're interested in working with somebody in Mauritius, you have an idea to say, okay, there are these people in Mauritius and this is what they're doing, and this is how I'm going to get in touch with them. So I think that is also very important, because we somehow work in silos, and we may find that we're doing the same work and duplicating the same results because we are not communicating, or it's very hard for us to have a platform to communicate.
Yeah, no, I definitely agree. And I think capacity strengthening in country is really, really key. And there should be a lot more of that, kind of supporting, in country capacity for sure. And I guess it's also kind of looking at coming from an ecology and conservation perspective. I guess I look at it from a, deforestation, of course, across Southern Africa has been a huge, issue. And so I guess I was thinking from I suppose looking at a One Health perspective, there could also be some really good lessons learned, like when you're showing, the kind of fruit bats foraging around homesteads, there's also those opportunities to try and actually help to restore habitats around, these really important, cave roosts, or like with Eidolon, really big colonies. So I guess, do you have any sort of, thoughts on, how your research can also actually looks like it can really feed into kind of conservation efforts too?
Yes, yeah, particularly when you look at the data that we're getting from the data loggers, it is true that actually even we have been going to the Lepers Hill Cave for, I don't know, for years now. And every time we go there, there's a new development along the way. We have got new houses, there's deforestation. Basically, we're seeing the area change. I don't know, I know that the area around the cave is supposed to be protected, so I really hope that we're not going to get people encroaching their homesteads up to right by the cave, but we are actually seeing that happening. And because the biggest thing is that people see bats as a nuisance. People do not understand why we should work to conserve bats. People will see they are smelly, they are in the ceilings, and things like that. Or people who fruit farmers will say they spoil their fruit. Then us pathogen hunters will say bats have pathogens. But at the end of the day, really, it's about knowledge. What is a bat? What does a bat do for the community? And how can we in the community help to keep the bats alive? Because if we do not keep the bats, then we are really disturbing the ecosystem around us. And like the work that we are doing, I think it's also exposure. So the people who know me and who know the work that I'm doing, and I talk to about bats, have some kind of an idea of the bats and why it is important to have the bats, even if we know that the bats have pathogens. So the information that we give, and we know we want to have this study over a period of years so that we see how it changes over time. As you said, you can see bats are foraging around the homesteads because they're going to the fruit trees in the homesteads. And how do we prevent this? It's also to if we have evidence to show people in authority to say, look, when you do this, these are the consequences. And then these are the additional risks to the population or in terms of disease transmission. So we need to think of the bats and make sure that the bats have the right habitat so that they stay in their habitat and do not come to the human habitats. So it's about information sharing, more research, getting the data, because even when you tell, even to drive policy, you need to have it backed by data. That's unfortunate. Even if you can see, what's going on, they'll ask you for the data. So it's very important to have the data so that we start having these conversations with people who can actually put a stop to deforestation in specific areas and say this area should be kept wild as a habitat for wildlife, not only bats. Because once you disturb one, you disturb everything. The whole ecosystem starts to crumble.
Great, thank you very much. So we have a question saying, amazing, thank you very much. Do you suspect an increase in viral shedding around the homes?
Not really that there's an increase in viral shedding around the homes, but the issue is that there is probably viral shedding during those months when the virus is active, when the virus is active in the bats. So what does that mean? So bats, when they forage, actually we tried, although it failed, we did put some cameras on some bats, but because of the size, we had to actually physically look for the cameras to download the data, and we never found them because we wanted to see the view of the bat as it's eating the fruit. What happens? Does the bat just bite from one fruit to the other? Does it drop the fruit? So even that dropped fruit, if it's contaminated with bat saliva, there's also a risk to anybody who comes to pick it up and things like that. So we wanted to answer that question really. But yeah, we did search and we told the local community to say, if you find such a thing, please give it to us. But we never ever found them. But it is just basically the risk is increased because the bats have come closer to humans. Ideally, bats and humans should exist, but should exist not close together. So the bats should be doing their own thing, the humans doing their own thing without really getting in contact with each other. But once the bat is in the homestead eating your fruit, then that is one step closer to you as an individual.
There we go. And so we've got another question saying, thank you very much for an amazing presentation. You stated that no viral genomes could be detected, which kind of sequencing was used to try and detect these viruses?
So we just use, so when we collect our bat samples, we use conventional PCR. So we have conventional PCR, and if anything is amplified, we then do sequencing. It depends. For short reads, we'll just use the normal Sanger sequencing. And then with these viruses that we detected, of course, they were first detected by PCR and then the sequencing of the short reads, the Sanger sequencing. But then to make the longer, like the different, when we're doing the phylogenetic analysis, we then used NGS for the longer reads so that we could have the full protein, for example, for the NP gene, and then able to do phylogenetic analysis. So basically, we use it different, depending on what we need, what the information we need to get, we use different platforms.
Thank you very much. So we've run out of time for any further questions, but I just wanted to end on this lovely quote from Iroro. So Iroro is a researcher working on African bats. She did her PhD in the US and runs an NGO in Nigeria. And so she says, an incredible talk, Katendi. Super jealous of your cool sites and movement data. Thank you for being a shining star for African women. So thank you very, very much.
Thank you very much, Shiro. Thank you. I appreciate it. And thank you, everyone, for listening.